won Fame Lab, not Malaysia Fame Lab, international. Of course, he won the Malaysian level first, and then he won the international level Fame Lab in 2016. Okay, so this is the MC for this day. Okay, just listen to this, all right? And then I hope you can identify the book and how the participant explain the scientific approach, scientific principle, because this is not a business pitching, and this is not a research proposal. So neither, it is not business pitching, it's not a research proposal, but it's more on effectively communicating a scientific principle of certain concepts. Yeah. So please listen to this first. Identify. Yeah.
It's not just pharmacists.
Okay. I'm not sure whether you can.
It's from the national.
Book. What kind of?
All right. Sorry.
Every out of time.
Yeah, let me.
So, based on my experience, I can understand. Okay, so the not Malaysia fan, international. Of course, he won the Malaysia principle of certain concepts. Yeah. So please listen to this first. Identify. Yeah.
Final participant, and it's not just a pharmacist. It's from the National Cancer Institute, Putrajaya, and currently pursuing his master's degree in analytical chemistry at University Putra, Malaysia. And I was just talking to him backstage, and he was telling me that he's also a trained lifeguard. But sorry, ladies, he's married. He's not ready to mingle, but he surely wants to tingle you with his science. So without further ado, let's have Sarif Naim Muhammad Ashar.
How are you to observe the t-shirt that he's wearing?
Picture is worth 1,000 words. If I take a camera and picture everyone in this hall, I'll be able to see beautiful faces, handsome, tall, short, and all the visible attributes. What if I take a special camera that is able to differentiate red guys from whites? In the branch of pharmacy, we produce radiopharmaceuticals for treating cancer. Radiopharmaceutical is a term combining radioactive and pharmaceutical. It is actually a drug, except that this drug is radioactive. So how does this radiopharmaceutical is used to treat cancer? Imagine that this is a pharmaceutical, which acts like a vehicle. It is chemically bound to radioactive material, which emits gamma radiation, forming a diagnostic radiopharmaceutical. It is then injected inside the body and travels towards the cancer cells. There, this gamma radiation penetrates through tissues and is detected using the gamma camera, forming an image. An image that is able to visualize, differentiate, and even locate tiny hidden cancer cells. Next, to destroy this cancer cell, the therapeutic radiopharmaceutical is produced by chemically binding the same pharmaceutical to radioactive material which emits beta particles. At a very short range, this beta particle will penetrate into the cancer cell, damaging it and also destroying it. This diagnostic and therapeutic radiopharmaceutical approach is termed as theranostic radiopharmaceutical. So what are the advantages of this theranostic radiopharmaceutical? With theranostic radiopharmaceutical, cancer treatment is personalized or tailored based on the patient's condition. Hence, it improves therapy outcome, predicts adverse effects, and also monitors therapy response. As a pharmacist, I'm committed in looking into different ways to treat cancer. My current research is in developing a radiopharmaceutical for bone cancer. And just like Thanos, hopefully one day, different type of cancer can be theranostically seeked and destroyed. Thank you.
Okay, right. They actually have around less than 2 minutes, I think, to explain the scientific principles of this. In this case, is targeted drug delivery, and they have to answer some questions. Okay, you can see these are the evaluators, the assessors, and they have questions. Okay, this is Arif, my former student. Okay, I actually groomed him for his presentation. Yeah, indirectly from this research methodology class. Okay, so he didn't win for the final, but I think he has given his best, and the one that win was a medical doctor who presented on drug what we call this, drug misuse or something. So you can actually, I'm going to give you this the whole this YouTube link, so you can watch. There are so many speakers, and you can see this guy is the one that wins. Let's look at him first a bit. Let me scroll back. Hey, just a moment, yeah. Okay. Okay. All right. Okay. Okay. Before that, I would like to introduce this MC is Dr. Siti Khairia. She won the International Film Lab, or FL, the International Film Lab, in 2018. I'm going to show you her video after this, okay? So now is the one that win this competition.
And with that lovely image fingering, let's give a huge welcome to our super singing shower singer actress, Dr. Hazrin. Woo!
Years ago, I was on call. At around 10 p.m., I received a call from the emergency department. There, I met patient X. He was psychotic, restless, and agitated. He was convinced that someone wanted to kill him. It turned out he had been addicted to methamphetamine for the past 10 years. Methamphetamine is also known on the street as stomac,
You can't hear me.
ice, or pill puder, literally horse pills, perhaps as a reflection of its effect of making you feel like a superhero. Sensation is with the drug is also known as psychosis. We treated him for about 2 weeks in our ward before he was discharged. 2 months later, he came back with the exact same problem. Sadly, this is a recurrent story for many drug addicts out there, because as many as 60% of recovering addicts relapse within 1 year. Individuals like patient X is just like any of us with jobs and family support. Because of drugs,
they are trapped in this vicious cycle of unemployment, poverty, crime, and broken families. It's easy to make one judgment against them. Are they stupid, careless, bad, immoral? Why don't they learn their lessons? Why are they repeating the same mistakes? Drugs like methamphetamine disrupt the dopamine activities in the reward center of the brain. Dopamine is what motivates us to repeat behaviors we find pleasurable. Dopamine is also important in the process of learning, which happens in the memory part of the brain called the hippocampus. Here, through the process of associative learning, the drug high moments will be linked to specific triggers. These triggers can be physical, such as hearing a song that reminds you of the time and place when you used drugs. Much like how the sight of 7-Eleven would trigger my 2 years old to cry for Kinder Joy, then gave us chocolate eggs with surprise toys that have been terrorizing parents everywhere. These triggers can be so powerful they induce cravings even if you have abstained for many years. The triggers can also be emotional, such as happy feelings, depression, anxiety, or something as benign as boredom. Much like how you would check your phone when you're bored, even if there's no reason to do it. By the way, I'm totally watching you guys just to see along. Finally, drugs will hijack the prefrontal cortex, and the brain loses its ability to recognize problems, find solutions, and inhibits disruptive processes. This is the reason why addicts like patient X continue to make terrible decisions, why they risk their lives to keep using drugs. Maybe we are too focused in cutting supplies and building our business with addicts. Maybe it's time for us to ask ourselves, are addicts criminals or victims of their businesses? How can we as a society collectively do better to fight this? Thank you.
All right.
Okay, so you can see that there are two different approach.
All right. So I'm just trying to show that in your presentation, for example, your proposal, your progress, or during your final presentation, the scientific principle behind what you are doing in your research is very important. And the more you practice delivering to the audience, the more you can tell a good story. So you can see there are a lot of examples. You can just watch Fame Lab, okay? And you can watch at international level because we have two candidates just now, one, Prof. Abimayu, and the other one, the lady, Dr. Khairia, how they present and they manage to win at international level. So these skills can be developed and it's not that difficult, but you have to practice. Okay, so listening to a good story, even though it's difficult because it's science, you can make it very interesting, just like what they have done. Okay, even though the terms are still correct, it's not like having different terms. They are all scientific terms, but we make it into a good story. So that is a very required. No, not that. That is a very something that you have to practice, and it is actually quite difficult. But you can do it. I mean, if you practice and learn the skill to deliver a difficult scientific principle or scientific explanation, but in a way that people can understand, even laymen can understand. Okay, that's very important skill for us scientists, because we need to be one day a science communicator. Because if not, then the science will just be in the lab. It's probably written somewhere in the journal, but if you are unable to communicate the science, then it's going to take a long time to be received by the whoever should receive the information. Okay, so communicating science is actually difficult. Apart from writing scientific papers, we also need to verbally communicate them, and it can be Of course, with all the jargons, it can be very boring, but we have to learn the skill on how to make it very interesting, based on the good skill of storytelling. Okay, so you can watch the film lab. It is in the YouTubes, right? So right now I'm going to show you, okay, so for those who just joined in, you need to watch this, another version of different speaker on AI assisted literature. Okay, and then you also need to watch this RSM, even though you don't think that you'll be using it, but it's a good data analysis. It's a good tools. Yeah, if you are working with a lot of factors in one time. All right, and I will try to get a talk on multivariate analysis. All right, so this one I will just stop share. So I'm going to share some example. Just a moment, okay? Okay, this is for those of you who are doing PhD, okay? You have to present for CE, yeah? CE is comprehensive exam, yeah? So this is an example of comprehensive exam. So it doesn't mean that you need to have a lot of results, because sometimes this you don't have that much. The CE has to be done uh, at least, yeah, on your third semester, I believe. Okay, so if you look at this, what I would like you to observe, of course, this is not, I'm not saying this is the best example, but at least it is pleasant. The presentation is pleasant. Okay. Okay, so one thing is about the title. Okay, it's not important for you to have the title finalized even at CE. You can always change the title even towards the end, but you must be well versed by during your CE that you know what you are doing. Okay, so in this case, you have to know what techniques that you will do and or you have done, and you confirm with that techniques, and for what purpose. Yeah, so because I'm doing this kind of work, so my example is more towards this kind of analysis. Yeah, right. So I'm going to show you why it's not moving. Okay. All right, so still in CE you have to do introduction. Okay, and make sure that you keep it interesting, and you keep it, you only have around 15 minutes to you only have around 30 minutes. Okay, so introduction is still needed, but then you have to focus on your results. Yeah, so what I would like to highlight here, the usage of diagrams, figures, photos are very important, because that helps the reader to understand better. Okay, so this is the problem, right? So using statistic are very good if you're talking about you highlight the issue, how difficult, how bad is the issue. So if you have like a new material to be developed, probably you can highlight a statistic of how many papers is right now publishing based on that material. Okay, so it should be less. All right, so these are all statistic, right? And this one is you highlighting the problem again and why you want to develop a new one. So in your case, probably you have different ways of looking at it. And what I would like to highlight here is again the usage of diagrams and figures. Yeah, right. Okay. The material also has to be highlighted. In this case, the bioreceptors. Okay, and then a bit of literature review. Okay, and when you do table for comparison, you have to highlight the one that important. Okay, so if you are working with material, then you highlight a series of material and what are the performance that you wish to present to the audience. Okay, because if you are working with solar cell, then different things that you want to explain to people. Okay, the parameters will not be the same. So you have probably different material and what is the parameters of the performance that you want to highlight to the audience. Okay, so problem statements again. Okay, so the usage of statistic, right? So objective, so I have told you the I think last week. Okay, you have to highlight if you're presenting progress in this case, because CE is actually a progress as well. You have to highlight which objective you are on. Okay, and telling people that you already completed 1, 2, 3, and these are the results that you are going to present. And let's say you still have 4 and 5 is fine because you have told the audience or the examiner that you only work until you have completed until 1, 2, 3. So experimental design, right? Right. So you have to show them clearly, based on the objective. Okay, what you have done. Is actually very easy if you present it based on the objective. Okay, so you can see that this student has difficulty because he has done a lot of work, but it doesn't, it was not successful. Yeah, you can see that he presented the initial plan and it was not successful after many times, and there are COVID issues. So you don't have enough data actually. So he presented another uh, alternative uh, proposal. So it's fine for you to do that during your CE if you caught with not less results. Okay, so just present whatever that you have done, and if it is not working, tell them if this is not working, and you have a contingency on what to do next. Yeah, so it's not always perfect. It's not always a beautiful journey. Just tell them the story, so you can see that there's not much results. Yeah, because a lot of fail experiment, but it's okay. Yeah, it's acceptable, but just tell them everything. All right, so you can see that actually there's not much results, but in the end, I have shown you this slide. Okay, in the end, he managed to pull everything and managed to get everything done. So this one I have shown you, but you can see the difference between the CE slides or the progress slide until the final. So don't worry if you like cannot don't have much results for CE. Now I'm going to show you other things. Okay, this is because I have shown you this one. Where is this slide? Okay. Stop share this one. So this is more on a bit of material which probably suits to many of you. So here is about the design, fabrication, and characterization of silicon nanowire. So it's about the silicon nanowire top-down fabrication. So again, that we start with introduction, because this one is on dengue. So this is a very big issue on dengue. You have this shock syndrome, okay? And if it can be fatal if you don't treat the shock syndrome. And then the issue of different kind of diagnostic method has to be used for the early infection and the late infection. So you can see the usage of pop-ups and the right font size. So it's pleasant to the examiners. The colors are nice, not too much colors, right? So you want to highlight about this strong demand, so make it a different color. And then the usage of general objective, probably if you wish, and then specific objective, and you highlight the keywords. And the keywords here that she wished to develop is the silicon nanowire base, and using the top-down approach method. And this is the approach, and this is the application. Yeah. So you play around. Remember that who is human being that going to evaluate you. Right. So this is step-by-step methodology. So let's look at right. And in order to explain the principle, okay, because in my line of work, sciences, we really have to explain a solid principle. So how it works. So same goes to you. If you justify on material A why it works, material B much better, then you have to specify. You have to tell about the science behind it. Okay. So I'm not going to discuss this, but I just want to show you that it is nice if you can, if you have the skill to do this. Okay, is yeah. I mean, while explaining, you can have different kind of. But I think you can do much better than this, right? So it makes the reader or the examiner understand better, and then it can appreciate your presentation. Yeah, the audience can appreciate your presentation much better. So these skills requires you to, if you don't have it, you have to learn, and you have to practice doing it. If you are good already, then it's okay. Okay, and the explanation of it. Okay, why, right? Why the size of the wire, silicon nanowire here, affect the sensitivity. So this one is very important because we don't want explanation like, okay, increase in size of wire and increase in reading. But what we are actually looking is the explanation as well. So put it in the slide. Put it in the form of diagram or figures. Yeah. Right. In here, it's easier for us to understand. The examiners, if we put it in the form of diagrams. Okay, this is the effect of DNA. And then you can see, because it involves a lot of instrumentation, so using diagrams and figures also can give you can give the reader more understanding. So this one is very nice for me, and I think you can do better than this. Step by step of the chemistry involved. Okay, so explanation. All right, a good pictures, a very clear pictures. Highlights. Okay. All right. explanation again, explanation of this graph is put down here. Okay. Mhm. Okay. All right. Okay. But information. So if you have like extra things to show, you don't have to put it in the presentation. Especially if you have detailed data for GCMS, for NMR, so you don't have to show it, but you can put it in the supporting information. And if the examiner asks, you can show the data. For example, here is the sequence. Okay, so you can put it in the supporting informations. Okay, so here another. I just want to show you another one, just one more, and that's it. Okay. Where is it? Oh, okay. Somewhere. Okay, so here is another this is more on different technique, but the usage of gold nanowires combined with silicon nanowires and gold nanoparticles. Okay, so these are all justification of the study. So there's a lot of words, but if you highlight a few that is very interesting, the one that you wish to tell the audience, then if you can put it in different color, then that's good. I would like to go straight to the graph. Okay, but before that, you can see that he did something like again general objective and then specific objective. So you can see the different colors, which is, because we're human being, okay, it stick to me that he wants to talk about DNA probe loading, electrochemical detection, and when he presented the method as well, he linked the objective together with the method section. Okay, so it's good. So easier for us to follow your presentation. And then results as well. You can see that the results are following the objective. So that is easier for the examiner to catch whatever that you're talking about. Yeah. The usage of error bar, right? And make sure that if you have a lot of a lot of curve to present, then you should use a very distinct color between the curve. Okay. Highlighted in red. Conclusion for first part, then right. Okay, so these are skills that you have to make sure that you can do as a researcher. It's not just lab skill, but you are to have this communication skill to communicate the science, science communicator. So not just verbally, also has to be in proper form, in slide form. And if you're talking about posters, now poster also has become in a form of slide. They are moving towards slide-based posters. So posters in Malaysia are still in a form of posters, normal posters like A1, AO. But if you learn to like prepare the slide properly, then the posters are almost similar. You know how to highlight your results and put it in a nice way in a poster. Okay, so after this, the next week will be your mid-semester break, and then the week after that we will start focusing on writing. Okay, first we discuss about scientific writing, and then towards the end we will discuss about thesis writing. And for writing literature review, I will try to get someone to talk about it. Okay, because writing literature, a review paper is very difficult. I seldom write review papers, so I don't have that skill to share with you. But I will try to get somebody who used to write review papers so that he or she can share with you the skill to write that. All right. So now you need to watch the videos, please. All right. So any questions before we end? Anyone involved in the convocation? I know Chai will. Fatin, are you involved?
Yeah, bro, involved.
So congratulations to all of you who involved in the convocation. Okay.
Thank you, bro.
Okay, thank you so much. Please watch the videos. Right. Bye.
Thank you, Prof.